Hi, I'm Rachel, and my group mates Mariah, Kristen, and I created this video about a breakthrough featured in a Scientific American article titled, "How Can Science Help Reverse Blindness?"
Teachable moment!
Macular degeneration, MD, is the leading cause of vision loss, and it's considered to be an incurable eye disease. MD affects around 196 million people. MD is caused by the deterioration of the central portion of the retina, the inside back layer of the eye that records the images we see and sends them via the optic nerve from the eye to the brain. The retinal pigment epithelium, RPE, separates blood vessels from the nerve layer and nourishes the retina. In macular degeneration, this layer is damaged. Retinitis pigmentosa, RP, is a rare disorder passed from parent to child that affects vision. As the cells on the retina deteriorate, the photoreceptors in these cells do not work the way that they're supposed to. What all cases of RP have in common is that when the doctor looks at the retina, they see a pigmentation that is caused by a bundle of tissue at the back of the eye.
Relates to class topic.
Retinitis pigmentosa is inherited, and some forms of the disorder are X-linked. If the mother is a carrier of this recessive trait, half of all offspring will be affected, regardless of gender. If the father is affected, female offspring will be carriers, and male offspring will be unaffected. Within the past few years, it was discovered that skin cells have the unique ability to be manipulated into almost any sort of programmable stem cell. Changing a single gene in a skin cell is enough to turn them into stem cells, or more accurately, pluripotent stem cells. The mutation is removed from these stem cells, and then 10 days later, cells are still mutation-free. Now let's describe the procedure that the researchers took. In this approach, researchers took a stem cell and replicated it many times, which turned the cells into perfect copies of the RPE layer that needs to be replaced in patients with MD. Those cells are then formed into a patch and inserted under the retina in the back of the eye to replace the damaged cells.
Teachable moment!
Induced pluripotent stem cells act like embryonic stem cells, even though they were developed from adult cells. Next, the CRISPR enzyme is used to remove the mutation related to RP. Because the stem cells are sourced from patients carrying the RP mutation, the stem cells will contain the mutation until it's removed. CRISPR technique is also used to activate certain genes, turning the stem cells into mutation-free RPE cells. To edit DNA, CRISPR cuts the molecule at a certain place, while a guide RNA molecule specific to the sequence being changed interacts with the DNA. The RNA reads the DNA and lets CRISPR know where to cut. Genes that are cut out are removed from the DNA and will no longer be expressed. In eyes affected by RP, photoreceptors die off. There are two types of photoreceptors: rods, which die off first, and cones, which die off in the late stages of RP. This loss of photoreceptors leads to a gradual shrinking of the visual field. Results of the study. After receiving the patch treatment, the two patients who participated in the study went from not being able to read at all, even with glasses, to reading 60 to 80 words per minute with normal reading glasses.
Teachable moment!
Here are our references, including our source article. Now for an inspirational quote to close.